So in my new role with Parks and Wildlife, I'm a big responsibility of mine is to help run their algae monitoring program. And really, I'm just kind of a point of contact for all our field people. And obviously, this is a growing problem, kind of a layup pun, but it certainly is a growing problem, something that the harmful algal blooms or algal toxins, that's a synonymous term that we use both. We're seeing it as a growing problem, partly because the growing season is getting longer as our summers are extending, which is the same reason the forest fire season is getting longer, but also as people become aware of it, more people are looking at the water differently, realizing that what they're seeing may be harmful algal blooms and not just regular algae. So it is a growing problem in that it is growing more, but also that people are more in tune to it. So we're seeing more people become aware of it and report it. And it's certainly something that has been making the headlines across the country. Harmful algal blooms have occurred on every continent, including Antarctica. And most countries have had some sort of problem with it. And in all 50 states, there have been some sort of problems with it. Luckily, in Colorado, we haven't had any dog deaths this year. That's kind of the most vulnerable population, but that's kind of what this whole program is geared to protect the public health and recreation, especially for us from the Colorado Parks and Wildlife standpoint on our park properties and state wildlife areas, but also just any, we're concerned with anybody who's going to recreate it to keep them safe. So before I dive into the nitty gritty, I'll just outline what we're going to talk about today. First, I'll just define generally what algae are. It's a generic term. And then what are the algal toxins that we're concerned about, how to detect the presence of algal toxins, and ongoing efforts to manage these algal toxins. So I'm guessing for Andrew and Mary, the latter half will be more informative, but sometimes just to explain to people what things are, sometimes it's just good to hear somebody else explain it. Maybe you just pick up different words to add into your explanation when you're out there on the ground. But so starting off with what are algae, really it's a generic term or general term for a diverse group of aquatic organisms. There's prokaryotes that are algae, there's eukaryotes that are algae. It's not, it's a diverse phylogeny of what we throw under that term. But really what we're talking about when we say algae, we're talking about not a plant. It doesn't have a stem or roots or leaves or vascular tissue. It's a simpler organism. But like a plant, they are the base of the food web and part of the primary productivity. And it's important to emphasize to people they are a healthy part of the aquatic ecosystem and they are a necessary part. So even though they produce toxins, some algae produce toxins that cause us harm, they are part of the ecosystem. It's not something we're trying to eliminate. We're just trying to manage and learn to live with it. Often we describe the algae by where it grows. So on the bottom left of the screen is a picture of planktonic algae. So the floating, free floating, or benthic algae that it's attached, or periphyton in a stream. But we kind of categorize it as it's either planktonic somewhere in the water column, or it's benthic and it's attached. And both can be forms. Cyanobacteria can be in both forms. So
Ashley, I'm sorry to interrupt. We are seeing still your first slide.
Oh, really?
I think we're supposed to see maybe the third one at this point.
Yes.
We see like the main PowerPoint slide. Sorry, I didn't catch that earlier.
Oh, I'm so glad you interrupted me. Oh, you've missed some really good stuff. Okay. Okay, so, oh, that's so weird because I'm seeing it. Okay. Oh, now if I keep it in this mode, are you seeing the second slide now of headline?
Yeah, we see slide 2. Thanks.
Okay, thank you, Megan, for interrupting. Sure. Oh my goodness. Well, now you can see my great material. So these are the headlines that it is a problem that is growing. Here's my outline that I just talked through. And then this is where I was just leaving off describing what they are. But this bottom left picture that you can now see is the planktonic algae versus the benthic attached algae. So sorry, I'm just going to leave it in this mode so you can see the upcoming slides, but it also allows me to flip through. So sorry, it's not ideal, but I can narrow this maybe. So these are just some different types of freshwater algae that fall under that generic category of algae, but they're very diverse organisms from one another. Green algae is a very common form of algae that's out there floating in lakes. It can look like lettuce. It can be stringy and filamentous, but it also, for you high country people, it's the same algae that grows on snow. So sometimes on the Continental Divide in the summer, you'll see this pink, what looks like dirt, but it's actually pink form of green algae. And that's actually a pigment that it produces to protect itself from the sun that's intense at high altitudes. So all, algae that you are familiar with and have seen. And then other things that can look like algae, although they don't in this picture, these are diatoms. These are also primary producers and photosynthetic organisms, but there really can be phenomenal variety of organisms. People make art with them because they have the silica shell. But when they form in mass, they can look like cyanobacteria, but they're not. And you won't know that until you either get a handle on looking for cyanobacteria or pull it under a microscope. But the diatoms can look like an algae bloom also. And they grow at the same time as because they're primary producers. But we're concerned about what I'm going to talk to you guys today about is the cyanobacteria. These are the group of algae that can form the toxins. And they are an ancient organism. They used to be credited with forming the oxygen in our atmosphere. Now there's some debate whether they came first or not, but they are one of the first photosynthesizing organisms. So as much as they can be a pain to manage, they're obviously an important organism. And then you're going to see lots of these kind of gross pictures today. But the bottom right one is Lake Erie, where Lake Erie is one of the largest lakes to have a cyanobacteria problem. So cyanobacteria is the more scientific term for them. Often people refer to them as blue-green algae, but that's, again, common name for the scientific name. Some species are capable of producing toxins. Not all species of cyanobacteria produce toxins, but over 50 species do. Their optimal growth rate for most species is at higher temperatures, so you do tend to see them in the heat of the summer more often than early on in the spring, although you can get a spring bloom when there's nutrient runoff, but they really will thrive in warmer temperatures. And they're able to outcompete other algae at high nutrient concentrations. So they can outshade other organisms and really form a mass at the surface of the water. And one of their evolved abilities comes from having these aerotopes, which are these gas vacuoles that regulate their buoyancy in the water column. And this gives them the ability to move between the bottom and the surface. And so they are able to regulate their movement in the water column. And this will become relevant to you in a moment, and we'll circle back to that. So some of the cyanotoxins that we're looking for, we don't find them all in Colorado, but this is the list of the cyanotoxins that cyanobacteria can produce. So microcystins is the most common one, and it's produced by most of the cyanobacteria algae are capable of producing microcystins. The microcystin attacks your liver, kidney, and it's a reproductive toxin. And it's the most common one when we have a bloom. Most likely, if there's a toxin involved, microcystin will be there. Cylindrospermopsin is very toxic, even more toxic, but it's less common, and it attacks the kidney and liver, so we call that a hepatotoxin. And then the others, the anatoxin and saxitoxin, are both neurotoxins and quite rare. The saxitoxin is the one that will get talked about if you are from a coastal state that eats a lot of seafood, or like the Chesapeake Bay. They will find saxitoxins in some of their shellfish. So that becomes more relevant as a food. People will be exposed to saxitoxins more when they're eating seafood. if it was growing in a toxic algae bloom. But the only thing we really see in Colorado are the microcystins. We're generally testing for microcystins, cylindrospermopsin, and anatoxin just to be safe, but we're always testing for microcystin as the number one. The deal with cyanobacteria is that when they are unpredictable about when they produce the toxin. You can have a huge, massive bloom, but not have much toxin, or you could have a very small bloom and the toxin would be present. So it's very unpredictable. And that is where a lot of the research that's happening now is to try and figure out what the triggers are, where the correlations are, so we could anticipate when a bloom becomes toxic, but we're not there yet and we don't know. Not all cyanobacteria species are toxic, and some of the very common ones are not toxic at all, but still form kind of a nasty looking bloom. And toxic species can have both toxic and non-toxic strains. So there could be the strain of the same species and it just happens to not be toxic. So when we talk about how we're monitoring and how we're testing, this will all come back to. And toxins are generally produced in warm, stagnant, high nutrient waters. So you're not often going to see these in rivers that are flowing because they get flushed out. You're going to see them in more eutrophic systems. They need stable water bodies. That's where you're going to see the cyanobacteria grow. So now I'm going to kind of go through some pictures just to give you ideas of what to look for when you're out there. These would be blooms that you would want to, if you saw something, you would want to report it to whoever manages that water body so that they could test it to protect people if, you know, and their dogs from drinking this water. So it can look really, this is a nasty bloom that looks like green paint. And obviously you don't want to touch it with your bare hands because you could then ingest it. And also it can also cause skin reactions. So we use gloves when we're really testing it, but these are web pictures when somebody's not. Sometimes it can look like grass clippings. This is a very common species that looks like just grass clippings in the water, but it's actually a cyanobacteria bloom. It can be clumpy, and where these single-celled organisms will then group together and form these kind of clumps and masses. And here's more of these kind of clumpy mats. This can be tricky because this is also what green algae can look like. So sometimes people will report a bloom as cyanobacteria that actually may be green algae because green algae has a very similar appearance in a water body. But you're seeing all of these pictures are kind of in these stagnant lake water bodies. And then the blue-green color. So the cyanobacteria gets its name because especially when it dies, many species, not all species, will turn this kind of turquoise green color that is very symptomatic and very indicative of cyanobacteria. It's unique to that type of algae. So that is a telltale sign. You won't always have it. So it's not something you can rely on to always be there, but when it's there, that's definitely cyanobacteria. But it can also, cyanobacteria can also look brown and be really diffuse and hard to tell. So really, whenever the turbidity or clarity of your water changes, you should look to see what's going on, because unless it's a storm that kicked up dirt, it's probably a bloom of some kind. So it's always worth testing. And so that's kind of, I'll hit home for Mary and Andrew. A new problem that now the EPA is trying to form a guidance document on, and we're just starting to look out for, is the benthic cyanobacteria that can form in the kind of littoral zone or shallow water of the lakes or really stagnant rivers. So it forms these mats on the bottom of rivers, and the mats can grow on sand, cobble, bedrock, wood. They don't care. They're just attaching to something, and it'll spread laterally across the substrate and can get very thick. When it's disturbed, like by a scouring or flow event, then the mats can detach from the substrate and travel downstream or float on the water surface. We haven't seen any of this in our rivers as of yet, but we're also becoming newly aware of it. It's less likely that it grows in really fast flowing water. This is more kind of a stagnant river condition. But these toxins, the signs of toxins, often what tells us that there's a toxin is if there's a dead animal or dead goose or dead duck reported, or God forbid, a dead dog that somebody reports. That often can be a sign of algal toxins. So animals are more vulnerable to it because they're going to drink the water. You and I are going to swim in the water. We might accidentally swallow some, or if you're water skiing, you might get a lot, but still not to the same volume that a dog or an animal is. And in some studies, they've shown that the animals preferentially drink from areas with cyanobacteria blooms, that there's something about it that's attractive to them. So this is kind of what domestic livestock deaths have been reported in Europe and South America and in North America. There's probably many more wildlife fatalities, and that goes unreported because we don't monitor them as closely, but the livestock have a value to them, and same with the dogs. It's a family member, so when they get sick, we hear about it. So that's who we're trying to protect, as well as humans. Some symptoms, if you were to come in contact with algal toxins, could include nausea, vomiting, diarrhea, skin or throat irritation. So that's why we say don't touch it with bare hands. You could have an allergic reaction or difficulty breathing. All of the neurotoxins that attack your nervous system, that controls your respiratory system. So it's one of these, get to a doctor immediately if you feel any of those symptoms. So here's kind of the overview of how our program works for monitoring for toxins. And we're always trying to iterate on this, but we first, in all of our water bodies, the lakes and state parks, as well as state wildlife areas and any other water body our people come in contact with, they do a visual assessment for the presence of cyanobacteria. So if you know what you're looking for by either Googling enough pictures or seeing it enough in your local reservoir, you know what to look for. If you see something, then you go test and grab a water sample and bring it to a lab. We can identify with a really kind of low magnification microscope what family if it's cyanobacteria and what family it's in. So it's not hard to learn to identify them compared to diatoms or green algae. And if we see it in a water sample in the microscope, then we test it. And we do 2 types of tests. And so this may be what Mary and Andrew will be more interested in. So we're just talking through our CPW monitoring program. That's a picture from a microscope of a cyanobacteria. So we have an administrative directive because we're such a large agency with people throughout the state. This is a directive that everybody can read and go to as like a manual. If you were just managing one body of water, I would come up with your own kind of sample plan or directive of when and what you sample and how often you sample. And that's really what our directive manages, is how often you're supposed to sample and how often you're supposed to do inspections. So in our areas that get a lot of, that get direct contact for water recreation, like swimming, paddleboarding, canoeing, water skiing, and wading, they do at least weekly visual inspections and for blooms. So they're from May 1 to October 1. Any lake manager is required to go look at the body of water. And I would emphasize that it's not always just out in the middle of the body of water. You need to look at the whole thing because it's often in these stagnant bays and coves. And for us, that's often where the swim beaches are, or where the access points are these kind of more protected bays and coves, because the cyanobacteria blooms can be very patchy. Another tool you guys may be interested in, this is a remote sensing tool called the Cyan app, and you can get it as an app on your phone. It's under development, and so it's still kind of clunky, but it's a partnership between the EPA, NOAA, and NASA to use both American and the European satellites to detect the cyanobacteria specifically. A lot of remote sensing satellites can detect plants and can detect chlorophyll. So you could see if you're having a bloom, but it could be the harmless green algae or diatoms. The Cyan app is able to pick up on the cyanobacteria specifically. And so sometimes for very remote locations, if we're not sure how it's doing or if the people are monitoring often enough, we'll log into this app and see what it looks like and see if there's a hotspot. And so this came from the Blue Mesa Reservoir area where there was in fact a hotspot in part of the reservoir. And so we saw that on the app and then sent somebody out. You better go take a look at that bay. It looks like the remote sensing tool is showing it. One thing to keep in mind with remote sensing is that it's a very coarse view. It's often on a 30 meter by 30 meter pixel at the very smallest. And so it may not pick up on every little bay, but it's a good screening tool, especially for remote areas, and a really cool tool that's under development and that these agencies are dedicating a lot of research funds to improve this. So this is where these are some tools you guys could use on just some basic monitoring before you get into the real testing with the strips and the expensive route. This is what we do first as well. When you go out to do a visual inspection, if you see something that you think is a bloom, there's some really basic tests that you can do that anybody can do. One is the bottle test, where you grab a sample of water in the worst location, where you see the bloom the worst, and you shake that bottle of water and then let it sit. And if it's cyanobacteria, eventually the algae, because of that aerotope and that gas vacuole, it will control its buoyancy and get itself back up to the surface. If it's green algae, doesn't have that, or diatoms don't have that, they sink. And so good things sink. The cyanobacteria is magnificently evolved and it floats. And so this is a good test if you can't tell the difference or you don't have, you just want to screen it first before you dedicate funds to testing it. This is a great one, and it's pretty fail-safe. Another one is the stick test. These are all both on CDPHE's website as well. So the bottle test and the stick test. If you put a stick in the water and it sticks like paint, like the paint images we saw earlier, and coats the stick like paint, that's cyanobacteria. If it hangs off it like strands, that's green algae. That's filamentous algae and is not cyanobacteria. This is actually a more evolved plant that is more complex than the cyanobacteria and is stringy and has filaments. But it looks gross, and people often report this because it's still a nuisance algae. It's still gross to fish or swim in. When you take a sample of water and look at it under the microscope, even at really low magnification, you can get good at identifying them because they're pretty straightforward. The same few species keep showing up, but these are our most common ones that I've been getting in samples so far this summer. The Aphanizomenon is the most common, and it's almost never toxic, though it can produce the anatoxin and cylindrospermatoxin. The Delicospermum is also very common, and that little green dot right there is its heterocyst, where it's able to fix nitrogen. So in lakes that don't have much nitrogen, it can still fix its own nitrogen and grow and bloom. And then the Gliotrichia is really fun and easy to identify. They look like little pom-poms, and both Delicospermum and Gliotrichia are able to produce this microcystin. And then microcystin is the one that the microcystin toxin, or microcystis is the one that the toxin gets its name from. It's almost always toxic, and it's producing the microcystin. So these are the ones we're looking for. If we see this in the microscope, then we go the next step and test it for toxins. And I'll come back to that. But so if we identify, if one of those species is present in the microscope, then we do this strip test. And you guys may already be using these and familiar with this, but this is kind of the same company that everybody is using for these strip tests. It's Abraxas, Eurofins Abraxas strip tests, and they look like little pregnancy tests, and it's about a 20-minute testing period where you just follow the instructions and add the pillow packets and add the stuff and let it develop, and then you see if a line is present or not. So if the line appears, there's 0 toxins. If the line is very faint or not present at all, then the toxins are very high. And they make these test kits, a different test kit for each toxin. So if you want to test for microcystin, you do one test kit. If you have the cylindrospermopsin and anatoxin, and they come in a box of 20 for $500, is about what it takes to get through a season for maybe one lake if you were testing every other week or something like that. If we get a positive on the strip test, we take it into the lab to confirm A, that the strip test worked, but B, to get at what levels, because different levels of toxin present, different concentrations will trigger different actions to protect the public. So these are some of the labs that are available. We use the CDPHE lab. They allow public to drop off samples as well. Not convenient for you guys up in the mountains, but you are, there are labs that can do this, and you can ship them. They're using either the ELISA or the LC-MS machines, and the ELISA is only looking for microcystin, which is the most common toxin. It's cheaper and gives the total concentration present, whereas the LC-MS gives you which microcystin congeners are present, as well as testing for the other types of toxins, but it's a much more expensive test. My one gripe about the lab testing is that it takes about a week. They run the machines once a week. And so they run it on Thursday. And so that'll give you all the samples that you've delivered up to Thursday. But then if you were to have a hit and positive bloom on Friday or Saturday, you're going to be waiting till next Thursday or Friday to find out what the levels are. So that may or may not be timely enough. But that's definitely, it's still wonderful to use them. And we have found that the ELISA machine agrees almost exactly with our test strips. But the test strips take like a trained eye, like there's kind of a gray area where if we go back and look, what you might call this color to this color is hard to tell. But a trained eye, the more you do it, if you have one person always doing it, then you'll get really good at it and used to it. And so that's what we tell our lake managers, is just have one person run the strip test, because that person will understand what to look for. And then our once we've tested, if we've had a positive, then we follow the recreational thresholds to trigger our response. So these are the EPA criteria, the microcystin at 8 micrograms per liter, which is the same as that parts per billion in the previous slide, or the cylindrospermopsin is 15 micrograms per liter. And then our state CDPHE, this is what we follow, the 8 micrograms per liter is the no contact advisory. So if we see a concentration higher than that, that's going to trigger a beach closure, no recreation until we've had 2 consecutive negative tests. And so if we have found, I'll stick on this for a second, if we have found the cyanobacteria are present, but the strip test showed no toxin present, that could change any day. The bloom could all of a sudden start producing toxins, or when the bloom dies, it releases toxins. So the water might look better because the bloom died, you had a cold spell, the wind picked up and it died. That doesn't mean you're clear quite yet. It takes a few days for that to clear out. And when it dies, it releases the toxins. It actually could get more toxic right at the end there before it gets better. So when we have a bloom present that's not toxic, we still post caution signs because it could change any day. And this is where the no contact advisory for humans is 8 micrograms per liter, but for dogs, it's 3. And so the yellow signs is almost a warning to dog owners that, hey, this stuff is present. We don't know at what levels. We don't think it's toxic, or it's below the human contact advisory, but that doesn't mean that it's safe for your pet. And so this just informs you what to do to get out and rinse with potable water. And this is a sign that came from the CDPHE, and we just attached our logo to it. If it's above that 8 micrograms per liter, that triggers a beach closure, no contact advisory, no even paddleboarding. Even though you're not likely to ingest a lot of water, you might have a skin reaction. At this point, we still allow people to fish, in the reservoir because that's a no contact activity. But I always want to see signs posted because I know I always bring my dog fishing and I would let my dog run around at the boat ramp or whatever beforehand. So I would want to know and want people to keep their dogs leashed up. And again, it tells you what to do. And we require, in our directive, we require that, again, these signs stay up until you get 2 consecutive negative tests. And if there's no cyanobacteria in the sample at all, you're clear, you don't have to post any signs. So that's kind of how our system works. Here's just an example of where we have detected toxins. This is actually, there's probably 3 reservoirs that are missing from this map, but these are kind of where we're not just seeing them in the Front Range or around the cities. We are seeing them up in the high mountains too. As you guys are experiencing with Willow Creek, it's not just coming from urban pollution, it's any kind of nutrient loading will feed the algae, will allow it to grow. And once it's in the system, it has a dormant stage. That gas vacuole allows it to sink to the bottom and it will go dormant over the winter. It is able to reappear next spring. So what we often see is once these reservoirs have had a positive, next year we anticipate these same reservoirs to have positives. In this case, 11 Mile did not have one this year. It had one last year, but we keep an eye on it because at any point that if the conditions are right, that same algae can reappear. But just a few more pictures and then we'll stop. The Cherry Creek Reservoir, just to give examples of what it looked like, people were recreating. A harmful algal bloom was detected, and it was well above the 8 milligrams or micrograms per liter of no contact advisory. As soon as we found it, they post signs, and these people are out of the water. Dewees Reservoir those clumps look bad. It doesn't look awful, but that number is awful. That 11,000 or 1,140 micrograms per liter. So that's a beach closure. Stagecoach in September of 2014 had very small amounts of microcystin, a little bit of cylinders spermopsin, and then a year later, the bloom has gotten worse. The conditions were just right, and it had its first real positive test for microcystin above detection limits and was 3 to 5 micrograms per liter, and then there are in warnings. Bar Lake is on the Front Range here. It's kind of at the end of the South Platte. So it receives a lot of urban runoff. And here we have that algae accumulates at the boat ramp at 82 micrograms per liter, but there was no widespread bloom in the lake. So the lake had to be closed because of this, because this is where people are accessing it. But the rest of the lake was clear and was testing negative. And then just to show you that it can develop and change in a matter of days. So August 6th to the 10th to the 13th, you go from having a bloom present that's detectable at a very high concentration of toxins on August 6th, then on August 10th, it gets way, way worse, although the bloom doesn't look horribly worse. And then on the 13th, it cleared up. And as it died, there was still some toxins present, but the algae looked much better. So they kept it closed for another 2 weeks until that bloom was they got 2 negatives in a row, even though it looked better. We look to the CDPHE for guidance there, the public health agency. We're just out there managing land. And so their website, they have a lot of resources on their website, that bottle stick test, the standards, and also their testing facilities. They also have FAQ and informational flyers. We try and educate our public that are using our resources. The EPA and CDC are the higher levels that are dictating kind of what the criteria are, and they're interested in always finding out where different blooms are happening so they can track it and find out what the new problems are. And then there's also different resources for recreational and drinking water professionals. We know that toxins, algal toxins, can leave the reservoir to downstream users and can be picked up in drinking water. Some drinking water treats it, plants treat it and kill the toxins, but most basic treatments don't. And so they need to be made aware that algal toxins are in their source water, as well as irrigation. It can travel in irrigation water. So we ask our lake managers, communicate to your downstream stakeholders. It's up to them to test their water and how they plan to use it. We can't report downstream forever, but you need to tell your downstream users so they know what's happening upstream, and they can have those strip tests and test it themselves or take it to a lab or make their own decision on how to use their water and exercise their water right. And then there's a toxic algae dashboard on the CDPHE website that includes map of historically impacted water bodies. And then questions and my contact information.